Hello everybody. Today we are going to discuss about artificial vegetative propagation in plant. We'll be learning uh, how to explain different methods of layering, describing various methods of grafting and method of tissue culture and its advantages. First of all, let us learn what is artificial vegetative propagation. As it is said artificial, so it is done artificially using vegetative parts of the plant. So if we get plants uh, from the vegetative parts of plant artificially, then this method is called artificial vegetative propagation, and it is especially applied in vegetable and fruit farming. There are three types of artificial vegetative propagation: layering, grafting, and tissue culture. So now let's discuss about layering. Layering, as you see in this picture, so what we do is we take the lowest most branch of the main plant and we keep under the soil. So that's why layering is method of getting new plant using the lowest most branches of the main plant. For that, the lowest most branch is bent and covered partially by the soil. Within 3 to 4 months, the part inside the soil, it develops roots and later it is separated from the parent body and grows independently. It is used commonly in plants like jasmine, rose, lemon, strawberry, etc. And there are 5 types of layering: simple layering, compound layering, tip layering, mound or stool layering, and finally air layering. Simple layering. In simple layering, the new plants are obtained from the old branch of the main plant by bending and covered partially with the soil. For that, we need to follow following methods. The tip of branches far up 20 centimeter from the main plant is buried under the soil. After 2 to 3 months, roots are produced, which is under the soil. Branches with roots are later separated and grow further in nursery, popular in citrus fruits like lemon plants. Compound layering. In this case, the whole branch of main plant is uh, buried under the soil, keeping its tip out. Later, from each node, new plants are obtained, which can be separated as a new plant to transfer and another place of the branch. And it is common in plants like apple, pear, lychee, etc. Tip layering. As the name suggests, in this case, the new plants are obtained by putting tip of the branches of main plant under the soil. Methods: tip of 5 to 8 centimeter of branches are put under the soil. After 3 to 4 months, the tip develops roots, and after that the branches are separated. It is popular in blackberry, strawberries, raspberries, etc. Mound or stool layering. In this case, the stem of plant is cut above 5 to 10 centimeter from the ground in the beginning of spring season. Once buds start to grow on them, they are covered with mixture of soil and sawdust, and then later roots are grown. In next spring, it is separated from the main stem. From the main stem, it is separated once roots are developed and then develops as a new plant. It is applicable in apple, pears, mango, guava plant, etc. Air layering. For that, any one matured branch is selected and 2 to 3 centimeter of bark is removed from it. This area is covered first with wet soil and then is made airtight by wrapping with plastic. In 1 to 2 months, the covered area develops roots, and after 1 month of growing roots, it is separated from the main branch to grow as a new plant. It is easy and very popular method among the farmers. Now the next method of artificial vegetative propagation is grafting, and it is the method of getting new plant by joining two branches of different plants. This is the part of one plant and this is the branch of another plant. So we get new plant by joining two branches of different plants. It is done before the plants bear new buds, and during the grafting, the root system that is used is called stalk, and the stem that is taken is called sign. So both sign and stalk are caught in special pattern so that they the combined layer of both parts are matched. So they should match in such a way that whenever they are joined together, they should match. The cut surfaces are joined by wrapping or tying tightly, and it is three types. It is done in three different ways: whip grafting, tongue grafting, and finally cleft grafting. Now we are going to discuss one by one. Whip grafting. This is the most common method of grafting, done especially in fruit plants. About 3.5 centimeters slanted cut is done in both sign and stalk. It is cut in slanted manner. After that, the cuts are brought in contact and they are held together tightly and then made airtight by wrapping with plastic. After 2 to 3 months, the cuts are joined completely. After that, plastic is removed. Tongue grafting. One of the easiest and most reliable methods of grafting done in Himalayan fruits. And for that, the double cuts of 3 to 5 centimeter are made on sign and stalk. So double cut means from the both side it is cut on sign as well as in stalk. After that, both the sign and stalk are adjusted, joining the wounds. At last, cuts are covered with the plastic and then tied with string. Finally, the grafted plant is left for 2 to 3 months to join the cut. The cleft grafting. So here, cleft is cut on stalk. On stalk, this is the stalk, the root part of the grafting. So cleft means deep cut is made on the stalk, and on the sign V-shaped cut is done. After that, both stalk and sign are adjusted together very tightly, and then it is covered with wax or tape to make it airtight. And after a few months, they join together, forming new plant. Tissue culture. It is the most advanced way of doing the artificial vegetative propagation. Here, the large number of new plants are obtained using a small piece of plant in artificial medium. Advantage is it is a large number of plants we can grow from the single small plant tissue in short time. Rare plants which do not have seeds can be conjugated using this tissue culture. Hybrid plants can be grown. It means that we can mix the gene of different plants and we can grow the new plant. And the plant with disease resistance can be grown. Similarly, new plants can be grown which have no viable seeds. Now, how to do tissue culture? Let's learn. First of all, we need lab with neutral artificial atmosphere where there is no any kind of other external factors, and plant tissues are taken and grown with plant hormone and nitrogen. For that, two hormones are used. One is auxin and cytokinin. If you want roots, then we need to put auxin more in amount, and if stem is required more, then cytokinin hormone is used more. But if both are required, then the auxin and cytokinin are used in equal portion, and in that way we can grow the plant tissue, and that plant tissue which is taken during tissue culture is called callus. The callus is cut into many pieces, and each piece is put in new media to grow further. Once stem starts to grow, we cut into many pieces, and on growth of roots it is transferred to greenhouse. Now, advantages of vegetative propagation, as it is practiced by farmers, so those plants which do not have viable seeds, or those seeds which cannot turn into seedlings, this method is very useful. So those plants which do not have viable seeds can be easily propagated using this method. Example, banana, rose, sugarcane. They don't have seeds, but we can grow new number of plants using this technique. The plant produced by vegetative propagation bear flowers and fruits earlier than those produced from the seeds do. So it means it is very much easy for the farmers to grow fruits and flowers, because using this technique we can grow flowers and fruits earlier than the one that we get from the seeds. It is cheap and rapid method of reproduction, so we can get new number of plants very easily. And lastly, it gives rise to genetically uniform population. It means to say that since there is no exchange of gene, so we can find exactly same kind of plants. It has disadvantages too. The plants that we get using vegetative propagation methods are short-lived and small compared to seed propagated by plants. No new varieties can be produced because here there is no exchange of gene. That's why we get just similar kind of plants, which is sometimes very harmful. And to do this vegetative propagation, we need skilled persons, and that is why it can be the expensive in comparison to seed propagation method. And finally, since here similar kind of plants are obtained as there is no exchange of gene, so these plants are very much susceptible to disease. With this, thank you so much.